Hello and welcome to this video PowerPoint lecture for biochemistry at Missouri Southern. This is Dr. Michael Garut, and in this first part of the video we'll be looking at an overview of metabolism of pyruvate and the pyruvate dehydrogenase complex. As you know, cellular respiration is the conversion of food molecules into water and carbon dioxide and the production of energy. We normally think about cellular respiration as being the choice of glucose, glucose going to make pyruvate, but then specifically acetyl-CoA going into the citric acid cycle with the electrons going through electron transport and then oxidative phosphorylation to produce ATP. This is somewhat arbitrarily divided into stages. Sometimes stage 1 is considered to be digestion of the food molecules, and what we're looking at here is considered to be stage 2. Nevertheless, the stage of acetyl-CoA production from food molecules, whether those be carbohydrates such as glucose, fatty acids, or amino acids, all producing acetyl-CoA, that's considered to be stage 1. Right now, we're going to look specifically at the conversion of pyruvate into acetyl-CoA. That step has no particularly good name. It's often called the bridge step or the transition step or simply the pyruvate dehydrogenase step. It is this step that happens between the end of glycolysis and the beginning of the citric acid cycle. The citric acid cycle then is sometimes called the next stage of cellular respiration, and then the final stage being the electron transport and oxidative phosphorylation. These two stages, the citric acid cycle and electron transport oxidative phosphorylation, take place in mitochondria. Here's an electron micrograph of a mitochondrion showing the folds of the inner membrane called cristae. A single one is singular, that would be crista, C-R-I-S-T-A, and cristae for plural. The reason that you have all of these folded membranes is to produce more surface area for the proteins that are involved in the electron transport pathway. Let's begin by looking at the pyruvate dehydrogenase reaction. This is an overall stoichiometry of the pyruvate dehydrogenase complex, which is an oxidative decarboxylation. It is oxidative because we are taking NAD and reducing that to NADH. So therefore, coupled with that is the oxidation of pyruvate, specifically this carboxylate group, to the carbon dioxide that is released. These are the cofactors that are required, and this reaction is energetically far from equilibrium, very favorable, and therefore one of the control points for entry of carbohydrates into the citric acid cycle. The pyruvate dehydrogenase complex consists of 3 different enzymes, but multiple copies of each of those enzymes, and these 5 coenzymes, which we'll look at. In E. coli, which is commonly studied, the E1, E2, and E3 complexes are present in these numbers, 24, 24, and 12. Each of those has a coenzyme or a prosthetic group that is attached to it in order for it to be functional. And at the bottom, we have the 3 overall steps of E1, E2, and E3. Step 1 being the decarboxylation, step 2, the oxidation, and step 3, the transfer of that to coenzyme A. Now, these steps only correspond to the first 2 steps of the pyruvate dehydrogenase complex reaction. The rest of it is to regenerate the enzyme into an active form so that the cycle can continue. Here are some of the cofactors. Coenzyme A has an ADP, adenosine diphosphate group, attached to a pantothenic acid group, and this beta-mercaptoethylamine group. This SH is the reactive thiol group. So commonly we will write acetyl-CoA as acetyl with an S, and then the rest of it being the coenzyme A. The lipoic acid, or the conjugate base of that, lipoate, is a coenzyme. Here's the lipoic acid on the left, and it is bonded in a condensation reaction to make an amide with the side chain of a lysine on the E2 enzymes. And that makes a linker that is able to take this reactive functional group and a long arm, which you can then fold around from one active site into another active site without this cofactor being required to dissociate and reassociate with the enzyme. And it cycles through these three forms: the oxidized form, the reduced form, and the acetylated form. Thiamine pyrophosphate is required for the E1. Thiamine comes from one of the B vitamins. Thiamine is a B vitamin. And the carbon that has this hydrogen attached right here, because of the sulfur and the nitrogen of the thiazolium ring, this hydrogen is particularly acidic. When that hydrogen gets donated, then that becomes a carb anion, which is the nucleophile that attaches to the carbonyl group of pyruvate. In the green, you can see the lipoic acid on the arms that are linking it so that it can transfer the acetyl group and back and forth between the E1, E2, and E3 active sites that it can fold over into. Here you see some advantages of multi-enzyme complexes. I'll let you read those. Here you see an electron micrograph showing the pyruvate dehydrogenase complex with the enzymes all connected together, so you can relate one of these with this picture. In a class activity, you are asked to write the chemical reactions for each of the 5 steps. You should be able to, for each of those steps, describe the reactants, the products, and the process. For example, in step 1, you could say that the substrate pyruvate reacts with the enzyme pyruvate dehydrogenase. It becomes decarboxylated while attaching the acetyl group to the cofactor thiamine pyrophosphate to produce the hydroxyethyl TPP. So that would be what happens in step 1. And you could then describe the other steps of that. In step 2, you're transferring the hydroxyethyl group to the lipoyl lysine while it becomes oxidized to an acetyl group. That simultaneously reduces the lipoyl lysine to the acylated form and the reduced form. Then you're transferring, in step 3, the acetyl group onto a coenzyme A, and acetyl-CoA is released as a product. Simultaneously, you have produced the reduced lipoyl lysine. Steps 4 and 5 are then required to reoxidize the lipoyl lysine so that it can continue to do step 2. In step 4, you oxidize the lipoyl lysine using FAD as the oxidizing agent. And then in step 5, you use NAD as an oxidizing agent to regenerate the FAD and produce NADH. In the E1 pathway at the top, you can see the reactive carbanion and how it is the nucleophile that attacks the pyruvate, and in this 4-step mechanism creates the hydroxyethyl TPP. In the second stage, you can see the pathway of transferring the hydroxyethyl TPP to the cofactor, the lipoamide, and then subsequently to coenzyme A. And then at the bottom for E3, you can see the pathway by which the oxidized lipoamide is regenerated. In this diagram on the right, the lipoamide group is shown as being attached with this linker that can therefore move back and forth between the places where that needs to react. So that needs to react at E1 when the hydroxyethyl group is being transferred onto it, and needs to react with E2 as that acetyl group is transferring to coenzyme A, and then it needs to react with E3 as the lipoyl lysine is reoxidized. So that linker arm needs to be able to move around to the active sites in each of the other subunits of the pyruvate dehydrogenase complex. These are more for trivia at this point than anything, but also to understand some metabolic disorders. So for example, arsenic poisoning works by this mechanism basically tying up this lipoic acid cofactor as this arsenite chelate and therefore inactivating the pyruvate dehydrogenase complex. In World War I, there was a treatment for arsenic poisoning, which was this dimercaptopropanol, which was somewhat bioavailable and then was able to accept the arsenite from this arsenite chelate and excrete that and restore the native version of the lipoyl lysine. The regulation of pyruvate dehydrogenase is obviously very important because it is a far from equilibrium step, and it is the committed step for pyruvate being used to generate energy as opposed to pyruvate being used to generate glucose. One of the things as a student of metabolism that you should definitely be able to do is to take each of the inhibitors or activators of each step and rationalize why it makes metabolic sense or why it would benefit the organism in terms of homeostasis to have the effect that it does. Here are a couple of examples. It turns out that pyruvate is an allosteric regulator of pyruvate decarboxylase. It is an activator, so this would be substrate activation. This makes sense metabolically because if the cell has an excess of pyruvate, then it needs to use that pyruvate in some way, and a natural way to use that would be to have it transfer the acetyl group to coenzyme A and enter the citric acid cycle to be used for energy. Similarly, if there is already a large amount of acetyl coenzyme A, then there is not necessarily a need for pyruvate to produce more acetyl coenzyme A, so it makes sense metabolically for acetyl-CoA to be an inhibitor of this same pyruvate dehydrogenase complex. The pyruvate dehydrogenase complex is also regulated by phosphorylation. So there is a kinase and a phosphatase. This could, for example, be protein kinase A, the same kinase that we saw being utilized in glycogen metabolism. And this could, for example, be protein phosphatase 1, again, that same phosphatase that we saw in glycogen metabolism. So let's look at ADP. If there is an excess of ADP, that would indicate that the energy charge of the cell is somewhat low, because you would like the energy charge to be high and the ATP concentration to be high and therefore the ADP to be low. Well, if the energy charge is low and the cell needs energy, you want this pyruvate decarboxylase to be active. Well, if ADP inhibits the kinase, then that inhibits the phosphorylation of the PDC into an inactive form, and so that is beneficial. You should be able to think through a logic like that for the rest of the activators or inhibitors, either in this diagram or in another one such as this. That's the end of the discussion about the pyruvate dehydrogenase complex, and in the next lecture we'll look at the citric acid cycle. Cheers.